Hi, this is Futurist Shara Evans, and today it is an absolutely fun interview that I've got lined up with Danny Cabrera, who is the CEO of BioBots, a company which is designing 3D printers that print biological materials. Good morning or good evening, Danny.
Yeah, I mean it's the evening over here. I know you're just getting started over there, but East Coast is it's pretty dark right now. Anyway, hey, yeah, thanks for that introduction, and yeah, excited to do this interview and yeah, get to talk to your audience.
That's wonderful. So let's start by talking about BioBots and what it does and how it even came into fruition. So what made you decide to do a startup company in the 3D printing space, much less in the biological tissue space?
Yeah, hey, so doing a startup sort of happened by chance, and being in the 3D printing space also kind of happened by chance. The biology space, that was much more that was much more purposeful. So I in school I was studying computer science and biology, and I spent some time working on synthetic biology. So really this idea that life is something that humans can engineer and use to do really incredible things. And sort of my undergraduate experience, spending a lot of time in and out of labs, I quickly realized that there were a lot of problems that were holding us back from being able to achieve the incredible promises of synthetic biology, including curing many diseases, eliminating the organ waiting list, even designing new organisms that could help us like suck carbon dioxide out of the environment, or even help us live on other planets. I don't know if you've seen Elon Musk's latest plans to colonize Mars, but the whole last bit there where Mars is spinning around and being terraformed, well, yeah, I mean, there's a lot of biology there. And working in the space, an undergrad for about 4 years, I quickly realized that there were two things holding it back. The first was that all of the tools that we had for doing biology were really designed around this core technology that came about 150 years ago. And that's the traditional tissue culture, so growing cells on a flat surface. So you get a monolayer of cells, which is great. It's great for doing experiments. It's pretty easy, but it has nothing to do with what's actually happening inside of the human body, right? This is complex 3D thing where you've got cells interacting with one another in 3D space, interacting with different materials. You've got signaling molecules floating around everywhere. It's a very different environment. So that's definitely one problem that I think was holding the field back. And the second is that all of the tools that we had, were pretty old. They were like pretty legacy equipment, and most of the work was actually being done by hand. So there wasn't a lot being taken advantage of in biology as far as automation, and even taking advantage of connected devices and Internet of Things type of work. So there was a lot of work being done by hand, which led to a lot of human error and a lot of irreproducibility. So I thought that started working on a 3D printer, thinking that this could be a good way to solve both of those separate problems. And initially, it was an academic project. It was actually my senior design project at Penn, and it was like a digital interface. So you interface with a 3D printer through your computer, typing lines of code, which seemed to be able to get rid of a lot of the manual work, since you had a robot actually performing the experiment for you, and also, it was able to build the tissues in 3D, which is literally an extra dimension on top of what we were currently doing. And initially, like I said, it was a project for school. But after I graduated from Penn, I was going to go get a PhD in bioengineering. But I had a summer off, and instead of taking the little money that I had left over and traveling through Southeast Asia, like I kind of wanted to do, I started just threw it down to just keep working on this project. And very quickly realized that if we wanted to keep working on this, we had to figure out a way to make money. And that's when the idea of actually selling the devices occurred to us, and started talking to a lot of the professors that I talked to while interviewing for graduate school. People were excited about it, and that's how we sold the first units.
Yeah, well, one of the interesting things about your background, Danny, is that you've got computer science and biotech and biology in your background. And it's actually something that I've been advising university students and high school students to do for a while now, is to combine those two disciplines. And I think that your idea came from the fact that you've got the engineering background as well as the bio background, and you thought about, how can I put this together to do things in a different way?
Yeah, absolutely. I wish I could say I planned it all, but it just kind of happened. I was interested in computer science, sort of been a coding geek for a while since I was in school, in grade school, and both my parents are programmers, so I sort of grew up around that. And it wasn't until I was introduced to biology that sort of a whole new world opened up, and that was in college. Just sort of stumbled into a drug discovery lab where there were. Where my job was to move around and treat hundreds of thousands of samples by hand, and I quickly realized whoa, man, no, we don't do it this way anywhere else in any other industry. Why are we doing like this in biology? It's supposed to be this high tech space. And anyway, it's sort of the result of that.
Yeah, it's great fun, and you've got a business partner as well who is your co-founder. Where did he come into all of this?
Yeah, so Ricky is our CTO. He was actually the one who was working on the mechanics side of things when we started building the devices. So he was working with a postdoc who was also a mentor of mine in the synthetic biology project that I was working on, and he sort of connected us, and that's how we started working together. Ricky was working on the hardware, getting the things up and running, and I was developing the software, and then we sort of transitioned where he's now running sort of the manufacturing side of the business, and I'm more on the strategy, sales, and marketing side.
So BioBots is now a commercial product, and one of the questions I have for most startup companies is, how did you get the funding to go to market, to take it from early prototyping into something that is a commercial reality?
Right. So we actually took it into commercial reality when it was still a prototype, which was probably in hindsight the right thing to do. So we had a really early prototype that we sold as our first unit. It kind of worked sometimes, but that was part of the sales, telling the person, hey, this is still a prototype, but if you want to work with us, we can make it work for you. And it's been a really good relationship with that. But it helped us, getting the device out into somebody else's hands. Number one, helped us validate that people were willing to pay money for this, and number two, helped us start seeing what people would actually do with it and what types of problems they were running into. And from that, we put together sort of a new device that was still a prototype. We actually called it our beta units, and we built 50 of them. We rented out about a dozen plastic 3D printers from a local makerspace, and had them running overnight. Then we found sort of unskilled workers that were unemployed here in Philadelphia, and paid them about $10 an hour to help us assemble the first units, and we put them in boxes and shipped them all over the world. And those were the very first units. So the funding to do that, to get even to that point, came from. The very early bit of it was from prizes that we'd won out of undergrad. By being an undergrad at Penn, I was able to win a few $1,000s that we put towards developing those first prototypes. And then from that, to really keep working on it, we went through an accelerator. So it's called Dream It Health, and they've been a great partner with us. Yeah, cool. So we actually we picked up our first board member from there. He's been extremely valuable in helping us grow and develop the business. Like you said, we were two engineers coming at this, and we had zero background in business. So having some trusted people that were on our side helping us think about how to turn this into a business was really helpful.
Yeah, it's essential, really, if you want to try to commercialize something.
Absolutely, yeah. And they really put in the first bit of cash, let us get those first prototypes out the door. And at that point, there was enough traction in the business. We were able to show that there was enough interest in the product. Even though they were just prototypes, we were still able to make some money off of them. And we showed that there was demand for them. And off of that early traction, that's how we were able to raise our first seed round. And it was a mixture of angels and early seed stage funds.
Okay, so let's talk about the BioBots printer. How does the printer work? What kind of materials are you using? What's the process that you use in the printer to make biological tissue?
Yeah, absolutely. So The printer sort of looks like a traditional 3D printer. It's a device that has an extruder. The extruder is moving in 3 axes, X, Y, and Z. And now what's interesting is that the extruder basically holds a solution of material, and that's they're different biocompatible materials that you can use. Obviously, different materials for different tissue types. Think about it sort of like how your inkjet printer at home has different colors, different colored cartridges for printing in different colors.
Great analogy.
We're trying to. Right. So we're trying to get to that point. In biology, we're not quite there yet, but that's basically the idea. You've got different materials for different cell types, and then you add your cells in there. So that's where the living part of it comes in. There's sort of no magic. And then the interesting thing is, the materials get pushed out of a nozzle using compressed air. So you pressurize the chamber, and material comes right through. And then there are a number of different processes that we can use to turn the material from a liquid to a solid. Really, the first one that we pioneered was we developed a process that used visible lights. As soon as the lights turned on, they would turn the material from a liquid to a solid without killing all of the cells.
That's important. You can imagine you don't want a bunch of dead cells.
which is what would happen if you use the traditional processes that are used in 3D printing, right? So plastic 3D printing uses like very high temperature to melt a spool of plastic. And then as it cools is when it turns into solid again. Obviously, you can't do that with cells because you'll fry them. You'll end up with nice toasty cells. And then the other option is using very high intensity UV radiation. And UV radiation is no good for cells. That's why, like, when you run out into a sunny day, the first thing you reach for is your sunscreen so you can protect your skin. So that's one of the processes at this point. We've got a bunch of other processes that we're using as well. Some chemical cross-linking, there's enzymatic cross-linking. There's a whole slew of them that we're supporting, as well as a number of different materials.
So in the 3D printing world with metals, I understand that some of the processes actually use glue, so they spray the metal powder and then they extrude the glue in the specific places. Is there a similar process that would work in biology?
Let me think about that for a second. I guess in some ways you can draw the analogy between the metal particles and the cells. So if you think of the cells as metal particles and the materials are different, sort of like think about a scaffolding material as the glue that keeps the cells together. That is kind of what we're doing, except that it just happens to look different physically.
Of course.
And but I think it's a similar concept.
So what you're really talking about is tissue engineering to be able to build different kinds of structures. And what sort of tissue have you grown or built in the BioBots printer so far? I'm not sure which term is actually the right one to use, whether they actually are growing or whether you're actually just printing them.
Well, so it's definitely a mixture of both. You start with printing it, and then over time they grow. So when you're done printing, you have a sort of a starting point where you've got a configuration of cells and material. But what's really exciting is what happens as those cells and that tissue really grows and matures over the course of weeks or even months. So then to answer your question, we don't grow too many tissues ourselves. We're really a technology shop. The core thing that we're doing here is developing the tools that are empowering other people to design and engineer the living tissues. So what I can do is talk about some of the things that our customers have been printing.
That would be great.
And it really runs the gamut. I think I'll talk about one that was recently published on Lab on a Chip, a great scientific journal. This was out of MIT, Harvard, Lee Kademasiani's group. They were using a BioBot to print a fibrosis model. So it's basically a 3D tissue that models thrombosis the way that it would happen inside of a human. So it's printed out of a gelatin-based gel. So think about it like it's a cross-linked collagen, and then they flow human blood through it. And the different the configurations that they've developed into this 3D geometry really mimics and models thrombosis the way that would happen inside of a human. And it's different than what you see observe in animals. So the value here is that these devices that are bioprinted can be used to test new drugs or new compounds and observe phenomena that you would miss treating on just a flat 2D 2D cell culture that you would miss inside of an animal test, but that you would see inside of the clinic. So if a drug is not going to be effective, if you're running a clinical trial, ideally you want to catch it as early as possible, hopefully before you get to the clinic.
So if I understand correctly, what they're doing in this trial is they're printing the tissue and then they're putting the blood, perhaps from a patient, through the tissue that they've printed. Is that correct?
Yep, that's right. So it's truly personalized, which is also what's really exciting about this. Every patient is going to react differently.
Yeah, wow, that opens up a very new area of personalized medicine. And I'll sort of jump ahead to a question I was going to ask you, but one of the things that occurs to me is that, a printer like BioBots, imagine going to your local doctor and, he or she scrapes a couple of skin cells and grows some culture, while you're sitting there and tests the different drug reactions on you before you even get the prescription and get the right dose. That would be pretty extraordinary. I guess we're a few years away from that, though.
We are a few years away from that, but there are definitely some of the larger pharmaceutical companies, more innovative ones, are definitely looking to bring that future to us as quickly as possible. And there are a number of challenges around not just the core technology to actually fabricate the tissues, but also the data infrastructure to handle all of the patient cells, handle all the samples, make sure that it's all protected. It's really important when we're dealing with patient data in this case. Especially because you can imagine what the world would look like if the data gets compromised and begins to be mixed up. So these are some of the things that these companies are thinking about, and we're helping them work through. So we're not just building the tools on the hardware side, but we're also developing the software tools and the reagents that are really empowering these larger companies to make decisions and move forward and bring that future, that really beautiful future, into the world as soon as possible.
What kind of time frame might we be looking at for something like that? Just, I know the future is not a set roadmap, but what's your estimate?
I don't know, and I definitely don't want to give the wrong answer. So.
We'll leave it for the future.
Yeah, we'll see when we get there.
Okay, now let me ask you about other kinds of tissues that can be grown or built in BioBots. You've done things like bones and cartilages as well. When I say you, I mean your customers inside of a BioBots printer. So how would one go about printing a bone, for example?
Well, I mean, nobody knows the best way to print really a real piece of human bone that can be implanted right back into people. But there are definitely groups working on that, and there, really the biggest mystery is in the biology. So how do you actually get cells to differentiate down the pathways they need them to, and then behave the way you expect them to. And so, as far as how to control that, the first thing is the bioink, or the material that you use to print that. And we've got a number of different materials that have different stiffnesses that are as stiff as bone, and you can sort of combine them with different signaling molecules to be able to control that. But the way you do it is the same as the way you do any other tissue. You'd have your solution of cells and material. You'd put it into the bot. You'd have your 3D geometry that you would have designed or grabbed from a CT scan or something, and then that 3D file would go straight into our software, and it would turn it into printer instructions. Then the device would go ahead, move in X, Y, and Z, deposit the material, and that would be your starting point. And then from that, you'd culture it up in an incubator or a bioreactor, and really let the biology or the cells take over. So like I said, that's the part of it that's still science. Nobody fully understands the way to do that, or the best way to do that just yet. But there are a lot of groups working on it, especially around bone and cartilage. It's a very hot area, and we're seeing a lot of great progress there.
Yeah, well, can you share any other customer stories? What else are your customers using BioBot for?
Well, we have customers working across pretty much every tissue type you can think of, ranging from bone and cartilage to skin. Maybe we just talked about blood, cardio. We've got people working on neural, liver, kidney, you name it. But even I think something that's a little bit different that we have some customers working on is working on plant cells. So printing different combinations of plant cells to build up new plants. And in this case,
sounds almost spooky.
Right. I mean, I think it's easy to imagine like the Super Mario plant, like chopping your but it's not. They're not thinking of going that direction. And at this point, it's really an art project. So it's sort of like showing the proof of concept that we can have new plants that are different. And I think the longer term implications are in using it for plant engineering. So things like food crops, that can building plants that produce more bountiful food, or more flavorful food, or even plants that are sort of friendlier for the environment. So you can imagine almond plants that take up less water from the soil, or that suck up more carbon dioxide from the environment. And I use these plants also as more than just a means for gathering food, but also use them for environmental engineering and impacting the environment in a very positive way.
Yeah, well, hopefully positive. Some of the darker sci-fi stories have these things running amok, and certainly it's a possibility. But as I look at outer space exploration or colonizing Mars, I can imagine that these sort of synthetic biology-based plants could be a really interesting way to go about terraforming a place that doesn't have its own living ecosystem. So
yep.
It's definitely an interesting place. And are any of your clients looking at printing organs at this space, like, a heart or a liver? I know those are complex organs, or maybe even something simpler, if there can be such a term for a complex biological structure.
Well, I mean, yeah, so, that's definitely a big part of the long-term vision of a lot of our customers, is to be able to use these tools to print much more complex 3D living things like organs. But everybody's starting small right now. The reality is that the state-of-the-art technologies the state-of-the-art groups are producing tissues that are a few millimeters thick. And these organs are an order of magnitude, if not more. If not towards the magnitude larger than that. So we definitely still have a ways to go, but people are taking the first small steps in our devices, and we're really excited to be empowering them along the way.
So how many BioBots are out there in research labs right now?
I can't give you the exact number, but we have devices in over 25 different countries right now, definitely all over the States, and we've been growing very rapidly since we started.
Oh, that's extraordinary. Your company's only about a year old, isn't it?
We're actually officially 2 years old.
Oh, 2 years
as of last month. So we've got like I said, we're on our second generation bioprinter now, which is pretty exciting. We've definitely learned a lot from our amazing customers.
And are you able to tell us anything about the price points of a BioBots printer?
Yeah, I mean, it's posted online, so it's very transparent. So the devices are $10,000 US. They're available at all times.
Not a lot of money. I mean, when you think about a research lab and the kind of equipment that they buy, that's actually quite small. And then you've got the bio-inks as well that you produce and sell to your clients.
Right. Hayden, I think that's definitely a big part of it. Is we wanted to build tools that were easy to use and cost effective. That way we could really begin to lower the barrier to entry to this type of work, to this type of research, so that we could motivate and empower as many people as possible to be working on this. Because it doesn't help very much if you have a great device that only one or two groups can afford. It's a lot more powerful when you've got a connected ecosystem of people working together.
Totally.
And, we're definitely excited to be building that community and building that ecosystem, and being providing the tools and building the technology that's empowering those people.
And one last question on what some of your clients are doing. I've been reading quite a bit about 3D printed cartridge that's used cartilage that's used in surgery, and bioprinted ears and things of that nature. Now, I know that most of your initial market are research labs. Are they doing any printing for actual surgical use on patients?
So, the patients in this case so far have been animals. People are definitely working to getting things into humans, but there are a number of safety trials they need to go through before doing that. And when people are showing off printed ears and pieces of cartilage like that, they're usually mostly for demo purposes, to really showcase to the promise of the technology and get people's minds going. I mean, it's the reason why you always see the ear is because we see ears every day, so it's something that we're familiar with. If you see an ear, you know exactly what it is. If you see something that looks like a kidney, you might know what it is, but somebody else off the street looks at it and might be like, what the heck is that? So an ear really gets people's imaginations going. However, not a lot of those are. I mean, really, none of those are being transplanted right now. There's nothing. There's no bioprinted FDA approved therapy just yet, but people are definitely working on it. So stay tuned.
Stay tuned and watch the space next year. Maybe some of these things happen pretty quickly. And
absolutely.
One last question for you, Danny, and that is, what is your marketing communication strategy? How are these companies finding out about BioBots? And I know your initial target market is research labs, but still finding them all over the world could be challenging for a tiny company.
Well, what's really beautiful about the space is that it is, number one, it's very distributed, so it is a very globalized world. Like, scientists work with each other regardless of where they are physically. But another thing is that they do meet up and they do talk to each other, and they not only talk to each other face to face, but they also read each other's papers. So if a group is using our devices and publishing papers with it, it doesn't take long for the rest of the community to find out about it. And likewise, when somebody's very satisfied with our product, and more than that, really with the people that they're working with, because we do more than just sell them a product. We really become part of their team. We really do empower them on the technology side and become the bioprinting expert in their lab. And so when people are very satisfied with that, they talk about it, and that's happy customers are replaceable.
Oh, so totally right. Referrals are the best and easiest way to get business. So I want to wish you every success in the BioBots venture. It sounds like you're on to a really great thing and a fascinating project. So thank you so much for taking time out of your evening to chat with us here in Australia and all over the world. And once again, thanks so much, Danny.
Absolutely. Thank you, Shara. Have a great rest of the day.